Hello everyone and welcome back to my YouTube channel. Today in this lesson we are going to understand the quality control in the ear. So the quality control, it is the how unfolded protein are degraded. So many proteins synthesized in the ear are rapidly degraded because they fail to fold correctly. Protein folding in the ear is slow and inefficient, and many proteins never reach their correctly folded conformation. So such misfolded protein are removed from the ER by process referred to as the ER associated degradation process. Okay, so in which what is the role of ERD? So ERAD misfold proteins are identified first. They identify the misfold protein. They return from the ER to the cytosol and degraded by the ubiquitin-proteasome pathway. Now, which proteins have sensor of misfold protein? So chaperones and protein processing enzyme in the ER lumen, which plays the important role into the misfold proteins. Okay, so how chaperone help to glycoprotein folding? The chaperone assists the glycoprotein in folding into its correct conformation. Okay, so as described in my previous video, ribosome synthesizes the polypeptide chain, and this polypeptide chain translocate within the ER. Here it is the polypeptide chain. It's glycosylation by the end linked glycosylation. That is the oligosaccharide binds to the asparagine residues in end position. Okay. Now the chaperone, they helps to protein folding. If the glycoprotein is correctly folded, it moves onto exit the ER and travel to the Golgi apparatus. Okay, here it travels to the Golgi apparatus. However, if the glycoprotein misfold, okay, you can say they fail to fold correctly. It is instead targeted to the ERD pathways for degradation. Now the misfold protein is transferred to the transmembrane complex with ubiquitin ligase activity. Okay, so this is a ubiquitin complex. It is a transmembrane protein also. Now ubiquitin ligase helps to the transfer the protein or translocated protein into the cytosol, from the ER to the cytosol. Now where it is ubiquitinated and degraded in the proteasomes. Here is the proteasome. They degrade the polypeptide chain. You can say the misfold protein, which is degraded by the proteasome, which is present in the cytosol. Okay, next, now we understand the ubiquitin-proteasome pathway. So the how protein degraded by the ubiquitin-proteasome pathways. Okay, in eukaryotic cell, the ubiquitin-proteasome pathways is a major mediator of regulated protein degradation. So first, here it is the ubiquitin. Okay, so ubiquitin first activated by the enzyme E1. This is the enzyme E1 which requires the ATP. Okay, now the E1 and ubiquitin. Activated ubiquitin is then transferred to one of several different ubiquitin conjugated enzyme, that is the E2. Okay, now next is the ubiquitin ligase E3, then associate with the both. Okay, they associate with the both E2. Okay, and substrate protein to direct the transfer of ubiquitin to specific target. Okay, so this is the target protein E2 E3. Okay. So main function E3, it is they binds to the E2 ubiquitin complex and protein target protein. Now the proteins are marked for the rapid degradation by covalent attachment of multiple molecules of ubiquitin, okay, to form the polyubiquitin chain. Okay. So here is the E3. Okay, they specify the ubiquitin to binds with the polypeptide chain, the specific site. Okay, now protein are marked for rapid degradation, okay, by the covalent attachment of multiple molecules. Okay, here it is the multiple molecules of ubiquitin to form polyubiquitination. Okay, they process the polyubiquitination and form the polyubiquitin chain. It is now the polyubiquitin. Okay, so now the polyubiquitinated proteins are degraded by the protease complex, which can be proteasomes. Okay, it is the proteasomes. Okay, here it is proteasome. This is present in the cytosol. Okay, they degrade the protein into peptides. Okay, and release the ubiquitin, which requires the ATP. Okay, next we understand the level of unfolded protein in the ER is monitored by the unfolded protein response pathways. Okay, so UPR, okay, unfolded protein response is activated in, okay, when it activated UPR, if an excess amount of unfolded protein accumulated in the ER. Okay, so this is a ER membrane, ER lumen, cytosol, okay, cytosolic side, you can say nucleus, nuclear membrane. Now there is the number of unfolded proteins, okay, the amount of excess, then and then the UPR, okay, UPR protein response is activated. Now the UPR leads to expansion of the ER, first expansion of the ER and production of additional chaperones to meet the need for increase the protein folding. Next, the reduction in the amount, also UPR leads to the reduction in the amount of newly synthesized protein entering into the ER. Now the unfolded proteins activated 3 receptors in the ER membrane. Okay, first one is the IRE1, ATF6 and PERK. So the first IRE1, okay, cleave pre-mRNA, okay, of transcription factor XPB1 mRNA. This leads to synthesize the XBP1, which translocates the nucleus, okay, which is translocated the nucleus and stimulate the transcription of UPR target genes. Now the second receptor, okay, ATF6. ATF6 itself is transcription factor, okay, ATF6. ATF6 cleave to release the active ATF6 transcription factor. This is also translocated to the nucleus and binds to the UPR target gene. Now the third receptor, it is the PERK. Okay, so PERK is a protein kinase that phosphorylates and inhibit the translation initial factor if EIF2, okay, and decrease the protein synthesis and reduction in the load of unfolded protein entering into the ER. Okay, it also results in the preferential translation of the transcription factor ATF4, which further contribute to induction of UPR. They also translocate to the nucleus, encoding chaperone, okay, the induction of the production of chaperone enzyme involved in the lipid synthesis and ERD proteins. So this is all about the quality control in the ER. This is all about today's video, and you will get materials of today's videos in the description box. If you are new in this channel, please subscribe and press the bell icon and share this video. I hope this content will be helpful. Thank you for watching my video.